Okay. So, hello everyone. I'm Aisha B. Rahman. I'm the chair of IEEE Women in Engineering Albuquerque section affinity group, and welcome you all to our monthly public talk series. We would like to thank Dr. Eva Chi, professor in the Department of Chemical and Biological Engineering in the University of New Mexico, for joining us as our key speaker. Thank you so much for making a time out of your very busy schedule. So Dr. Chi obtained her bachelor's degree, one in chemistry and one in chemical engineering from the University of California at Berkeley. She obtained her master's and her PhD degree in chemical engineering from the University of Colorado at Boulder. And now she is a professor in the Department of Chemical and Biological Engineering, where she's also serving as an associate chair and director of undergraduate programs. So thank you so much again. And I want to pass it down to Dr. Siropolo, advisor of our affinity group, to say a few words on behalf of our group.
Thank you, Aisha. Thank you for the kind introduction for our speaker today. It's really a great honor to have a colleague here today to give, I'm sure that she's going to give a very inspirational talk. I just read her short bio and I was impressed about her journey. So I look forward to your talk, Eva, and we can keep it quiet and the floor is yours.
Thank you. Okay, I'm going to go ahead and share my screen. All right. You're seeing this okay?
Yeah.
Okay, great. Thank you for the invitation. I'm happy to be here. And thank you for the kind introduction. So for the audience, I prepared kind of a presentation of the overview of some of the research projects that we're working on in my group at the University of New Mexico. And when it comes down to it, I think the theme of finding solutions to problems is what we're doing and what all engineers are really good at doing. So I want to show you kind of a few, maybe not final solutions, but on the way to solutions, both in health and kind of surprisingly in education that we've been working on. And hopefully it also shows the wide variety of problems that we can solve if we put our minds to it. So one of the first problems that we have studied for a long time is the case of bad proteins. And when I say bad proteins, I mean proteins that are in a form that they're not supposed to be. In this particular case, we're going to even narrow it down to bad proteins that start to form and accumulate in the brain. Some of the particular diseases that bad proteins cause, such as Alzheimer's disease, Parkinson's disease, Huntington's disease, etc. Some of those that we've heard about, they're pretty prevalent. What we know is that, for example, for Alzheimer's and Parkinson's disease, we don't know what caused these diseases, not particularly, even though we do know what we might find in really advanced disease states, but we don't really know what really caused it. And as of right now, there's no early diagnosis in that we don't know somebody is developing Alzheimer's and Parkinson's until they exhibit symptoms. And by that time, there's a lot of damage to the brain already. And obviously there's no treatment. There's no treatment to slow down the disease progression or reverse the disease progression. So those are some of the kind of the big picture problems that we're trying to tackle. I want to talk about specifically what we have done through the 15 years of research at UNM is that we have discovered a class of novel fluorescent molecules. One of the really good performing ones is down here, that can selectively detect the bad proteins. from the good ones, which is really important because we want to be able to pick out the bad proteins that cause the disease. And what's more important is that it could do it in a number of disease conditions. So not only for Parkinson's disease, but for Alzheimer's disease as well. So the idea of these molecules is that this is what the chemical structure of these molecules look like. What we do know is that they're very sensitive to their environment. For example, in water, these sensors are dark. When it sees these bad proteins, it's like a light bulb, they turn on. And because of this turn on mechanism, it makes them really easy for us to detect. So with these particular molecules that we discovered in solution, we have also shown that they work in brain tissue as well. So here are a select few fluorescence images of a wild type. So this is a really thin slice of brain from a wild type mouse. So this is healthy mouse. This is one that a mouse model that was created to kind of recreate Alzheimer's disease. This is a brain section from an Alzheimer's mouse, and this is from a human brain tissue. This particular patient had frontal temporal dementia, which is a little bit different than Alzheimer's disease, but it's caused by the underlying, the same kind of bad proteins. So each of these brain sections were soaked in a solution of our sensor. In the wild type mouse, where we don't expect a lot of the bad protein, we don't see many features. However, in the Alzheimer's mouse, we start to see these halo-like structures that are dyed green by our sensor. And this is exactly the characteristic bad proteins that we expect to see in Alzheimer's disease patients. Moreover, when we move to human brain tissue, our sensors are capable of picking up these bad proteins as well. And we've done a lot more characterization of the different type of brain tissue, not just from mouse and human, but also from rat Alzheimer's model as well. And we also co-stain these tissue sections with antibodies, for example, to confirm that these features are what we think they are. And all of this were just recently published in Biosensors. So in addition, what we're really excited about these proteins is that not only are they able to kind of bind to and start to fluoresce in the presence of these bad proteins that cause disease, they also have this special property of photosensitization, meaning that once our compounds are bound to the bad protein, we can shine light to it. It starts to generate singlet oxygens, which can cause oxidation to the bad proteins that they're binding to. We haven't been able to demonstrate this, but other groups have shown that if we can cause oxidation to the bad proteins, it can actually prompt the body to clear out those bad proteins. So this to us seems like a viable therapeutic pathway. And the reason why our compounds, we're excited about our compounds is that our compounds are actually selective. So they're not going to go after the good proteins that we all still want in our brains, but because they're selective, they can selectively cause the clearance of the bad proteins. So what we envision is that we have these bad proteins that are misfolded and aggregated in the brain. With the technology of our sensors, we can potentially lead to the early diagnosis of these proteins, which as of right now is not possible. And if we could also demonstrate that these turned on sensors can also cause oxidation, which we know it does, if the oxidation can also cause these bad proteins to be cleared away from the brain, perhaps this is a compound that can do both. It can detect and it can destroy. So that's kind of this is the direction that we're going. And hopefully this will lead to more fulfilling, healthy lives, particularly for the elderly. The second engineering solution that I like to talk about are these UV activated polymers and oligomers. They're actually from the same type of molecular architecture that we have shown recently to be very, very efficient antiviral materials. For example, against the SARS-CoV-2 virus that is still causing this pandemic. And this is in collaboration, as is the previous project, a collaboration with my colleague Dave Witten in my department. And he's actually holding one of these compounds that glow under the light. It's not the same compound as the sensors, but it's from the same family of compounds. So this project came about, if we all remember, a few short years ago. COVID-19 was spreading like wildfire, and we shut down the state. All of us went on Zoom for classes and everything else. So while we're busy adopting to Zoom Live, us as researchers, we're also thinking to ourselves, oh my gosh, what can we do to contribute to combating this pandemic? A few of my colleagues jumped into action, such as Professor Heather Canavan and Professor Christina Salas. They started 3D printing masks that holds filters, for example. Professor Canavan took the 3D printer home. This is her garage with her students. They were printing things like the ear savers for nurses, and other parts that would make, for making masks. So, while, I learned about their efforts when we're meeting on Zoom, it also got us thinking, Dave and I, what can we do? And that's when we thought, oh my gosh, we had a study that we published about 10 years ago that showed a class of compounds that we had been studying for antimicrobial purposes, killing pathogenic bacteria, that they're also effective antivirals. At the time, we tested kind of the model viruses that doesn't actually cause human diseases, but we did test those 10 years ago, and they were shown to be very effective. So what we thought was that, do these compounds, are they also active against the SARS-CoV-2 virus that's causing the COVID-19 pandemic? So very quickly, we assembled a team with Dave, Lene Ista at the Center for Biomedical Engineering, who has a microbiology material science background. Critically, we found a colleague at the North Campus School of Medicine, Alison Kell, who is a virologist, and she actually works with the SARS-CoV-2 virus. So we got together and said, would you be able to test some of our compounds? Let's find what we have in the lab, and let's see if they're active against the SARS-CoV-2 virus. So these are some of the compounds that we found. These were literally in test tubes in the drawer of Dave's lab. And we selected 5 of them for this initial test. 3 are what we called oligomers. This is their molecular structure. And 2 are polymers. So these are larger molecules with n number of repeat units. So we made solutions of these compounds, kind of, this one is dark, and they're kind of yellow to red to orange. We made solutions of these compounds and passed them on to our collaborator, Alison Kell. And what we wanted to know is that, can these oligomers and polymers that we had shown in the past able to inactivate bacteriophages, which are viruses that affect bacteria but not us, can they also interfere with viral activity of the SARS-CoV-2 virus? So, so Allison took the viruses and tested them. The way that she tested them was basically by visualizing the viruses. The viruses are too small to look at, and so this is a neat set of experiment. We saw them by counting this plaque assay. So what she did was she grew human cells to a layer in this dish, which is about, I want to say, 5 inches wide. And once the cells are grown, she exposes the cells to a solution of the virus, or the solution of a virus that's treated by our compounds, and then let that sit for 72 hours. And then she adds a dye to the solution, so you could look at the cells. The live cells is dyed blue, and then she counts the number of holes that's in this layer of cells. And the reason why there's a hole is because there's an active virus that had killed the cells. So the cells are not there anymore. So each, what we called a plaque, correspond to one single viral infection. So, you know, there's a lot of, anyway, there's more to the experiment, but the bottom line is we can now visualize how many and quantify how many viruses are in a solution based on how they were treated differently by the different compounds. What we found is that when there's no oligomer, so this is on a log scale of plaque forming units per milliliter, or the number of active viruses per milliliter. And what we see is that in the presence of no oligomer, you would have to, and this is light activated, so we put this in a light chamber, which I'll show you later. What we saw was that the viruses are quite robust. You would have to shine UV light on it for 60 minutes to kill some of the viruses. But when we look at in the presence of our compounds, this is oligomer 1, 2, and 3, we show that very quickly after. So because this is a log scale, I just want to, you know, kind of, so originally untreated viruses have 5 logs of concentration. 10 minutes after treating with oligomer 1, it dropped down by 4 logs, almost 4 logs. So what translating in terms of percentages, this means that 10 minutes after treating the virus with our oligomer 1, 99.99% of the viruses are killed. And after 15 minutes, basically we have 6 logs of kill. So oligomer 1 performed very well. Oligomer 2, similar. Oligomer 3 similarly reduced 4 logs of kill after 5 minutes, and then nothing was detected after 10 minutes of irradiation. Oligomer 3 didn't work as well. We saw similar results from the polymers. And after we had performed the first study, what we thought was, okay, so this is irradiated under, you know, kind of in the UV region, not deep UV, but in the UV region. So in order for these compounds to work, you really need to have a UV light. Since then, a very talented synthetic chemist in Dave's group started making more compounds to, you know, that absorb under white light. So fluorescent bulbs or room light that we would use typically in an office building. And this is the result. So this is what the light chamber looks like when the solutions are being incubated. This is what we have in our lab. In Allison's lab, she also has one, but it's in a biosafety level 3 lab, so it's isolated. But what we see is that, again, this is the virus on its own, and this is 5 minutes after irradiation with white light. we see 3 to 4 logs of kill inactivation after 5 minutes of irradiation, and we didn't detect any viruses longer than 10 minutes. So what we have shown in this study is that these compounds, again, are very, very effective at inactivating the SARS-CoV-2 virus, and it does so very quickly under the irradiation of white light. We're really excited about these compounds because of what we know about their mechanism of action. The question is, how do they inactivate the virus? How do these compounds also kill bacteria and different viruses? We've tested 3 different viruses so far. From what we know of these compounds from lots of other studies is that they really have a non-specific mechanism of action. They don't block a single biochemical pathway, for example. They do so by dissolving and disrupting the cell envelope or the viral envelope, which is what we're trying to study right now. But this non-specific broad spectrum mechanism is exciting because these compounds could potentially be effective against a broad spectrum of viruses. Unlike the vaccines that we get, where each time the virus mutates, we need to have a booster shot because otherwise it wouldn't be effective. These type of our compounds, being also a material, all of a sudden start to look like they could, because they're capable of killing lots of different viruses and bacteria, they potentially could be great materials to use to combat not just existing but emerging viral outbreaks as well, because they're non-specific. So some of the applications that could potentially from an engineering approach include, for example, filters in ventilation systems where they capture the virus, and if there's light on it, it inactivates the virus. Longer lasting wipes, in that if you spray ethanol on a surface and wipe it, it's very good at disinfecting the surface. But once that alcohol evaporates, the surface is no longer is no longer sterile. Whereas because these molecules are not volatile, they could be woven into wipes, they could be incorporated into fabrics, etc., and they're long lasting. So potentially they could be they have applications beyond what we use right now. And also they're superior because of their light activated activity as well. So these are the disease detection of Alzheimer's and Parkinson's, and also the pandemic. Those are kind of the 2 problems that we've tried to engineer solutions for. Another one that I want to talk a little bit about is that in my experience as a teacher teaching chemical engineering courses, and I think that's just how engineers naturally think. We're always thinking about how do we do this better. And the same goes to teaching. I've taught chemical engineering lab for a few years, and I have to say there are many things that I tweaked and changed. But one of the things that we always think about is, engineers, the undergraduates spend 4 years, around 4 years with us. And we're not just teaching you book knowledge. We really want to train you into good problem solvers. And the idea is, how do we do that better? And one of the things that we've done is in the department is to empower students to solve real problems. Oftentimes the curriculum have students take all these theory classes, And when you get to the senior year, you have your senior capstone design. And that is when, oh my gosh, I need to, for the engineering students, they need to design a process to produce a particular product. And that's when they really put all the knowledge together to solve an actual problem, from a prompt that the professional societies put out. So what we thought was that, we should put those problem solving design for solutions earlier on in the curriculum. So in our department, we've put design challenges into all levels of our classes, and to do it in such a way that it's not extra for students. It's not just an extra project that you work on, but you kind of weave those projects into the material, into the class. And this is one example that I did, which I had fun doing it, and I think the students liked it as well. So in 2020, and this is prompted by necessity as well, because that was when everything was still shut down. I was teaching a senior chemical engineering lab class, and I thought, oh my gosh, how am I going to teach a lab class, where students get to go into the lab and, do something. So instead, I talked with a few colleagues, and we basically redesigned the entire course. Instead of handing a recipe to the student and say, go into lab, move this dial to this, put this setting to that, and do this experiment, collect this data, do this analysis, and then report back to me what you did, which is how chemical engineering experiments have been taught for the last 30 years. We thought, okay, we have this distillation experiment that we typically do. I'm going to ask the students to design their experiment. Moreover, I'm going to make it relevant to what they're experiencing right now. So in 2020, they designed and carried out a distillation experiment to produce hand sanitizer, which is exactly what distillation does. And to make it even more relevant to the student, the solutions that they sought out came from the community, and the solution was for the betterment of the community. So they visited places in their community, in this case, the breweries, to find their source of ethanol to make their hand sanitizer. And they worked in teams, and they also decided on where they're going to distribute this hand sanitizer that they make. Some of the students chose shelters, prisons, schools, etc. And they figured out how much they need and how much they could produce with the lab scale distillation column that they had. And they went ahead and did the experiment. And then they figured out, okay, were they successful at making hand sanitizer or not? And this is kind of a mock-up bottle that were made for the students. So this is where they can put their product. It went really well. So how the class was taught was written up in the Mirage magazine, which is a UNM magazine, in the spring of 2021. And I told the reporter, just come to lab, pick a day when the students are there, and some of the students were interviewed. The students thought, even though it was a struggle, they really had to figure out how to do things, right? They're not just, again, given a recipe to do something in the lab. They really had to think about, okay, what product am I trying to make, and how do I get there? So I think their learning experience was also deeper and more meaningful in that case. But what was really good was that, with all the Zoom classes that year, they really liked it, and they thought the experience was very motivating, because they can actually see my effort in the lab could actually be used for something. So that was very nice. So this is one example of what I did in a particular class. We're also working with Vanessa Shvila, is the learning scientist that our department has been working with to implement all of these design challenges. And we also reached out, or actually they reached out to us because they heard about some of the Stephanie Wetstein and Jennifer Brown, two chemical engineering professors from Montana State University. When they heard about some of the education stuff that we were doing, they reached out to us and said, hey, we have some of the similar challenges in our curriculum. And they're also actually doing lots of innovative teaching stuff, education. So we started talking, and then we said, let's work on a project together. So we brainstormed through lots of meetings and, really, kind of empower the students to solve real problem was, kind of give this idea of, hey, let's look at agency in chemical engineering experiments. We picked the title because it's a nice acronym, ACE, because we recognize that experiments are really critical for preparing future chemical engineering students or any engineering students, because experiments are needed to test and support ideas, to assess safety, to optimize processes, and to test solutions, not to mention research, and kind of investigate unexpected things. The idea that, the scientific process, right? You come up with a hypothesis and you come up with a way to test it. That is practiced by engineers all the time. So our idea is, how do we prepare them better through redesigning what we do in the lab? So the central question, and this was just funded, the central question that we're trying to ask is that, how does having agency meaning where students can make consequential choices, not just what background do I want to use for my presentation, but real meaningful consequential choices about what they want to make, how they want to design their experiment, what data do they want to collect, and how do they want to communicate the finding to other people. How does having agency in many different areas with performing an experiment impact the learning and the professional identity formation of engineers? The idea that, how students feel about themselves as engineers. And, to kind of drive the point home, this is an experimental setup of a picture of it in a brochure for our department from 1978. This is what the experiment looks like today in the lab. So a lot of things haven't changed about how we do things. So we're trying to look at how, some of the electronics have changed, given that, but what can we do to fundamentally change how we do these experiments, how we have student involvement in their own learning, how does that impact their learning, and how they feel about themselves as problem solvers and engineers. So this is, what time is it? This is all that I have prepared. Thank you for being here and your attention. And I also want to thank the many high school students, undergrads, graduate students, postdoc collaborators, and funding agencies for supporting our work over the years. I want to leave with this last slide and say, go forth and be awesome engineers. There are lots and lots and lots of problems to solve. And we have all the skills to do it. I probably should have paused and asked for questions earlier on, but any questions?
Thank you so much, Eva. I love the last slide, actually. It's very, very inspirational. Okay, so Aisha, the floor is yours. We can open the room for discussion and questions. It's not really necessary to be questions. They can be just discussions.
Discussions, exactly. So if anyone has any question, you can raise your hand or use the chat as well. I'm going to ask the first question. So I saw that you had the 2 bachelor's degree, one in chemistry and one in chemical engineering. So I want to ask you, what made you first decide that you want to be an engineer? Because after that degree, I see that all of your degrees are on engineering.
That's a great question. So I started off in chemistry, because when I took my first chemistry class in high school, and I just felt like the kind of the world opened up, because all of a sudden I can see, wow, I can really understand kind of everything that's going around me, which I was always curious about as a kid. So I started off in chemistry, and at the time, first generation college, for my family, I really was not thinking about graduate school or anything beyond a bachelor's degree. So at the time, when I was in the chemistry program, which I loved, I thought, oh, okay, so, how does my job prospect look like with a chemistry degree? And it didn't look very good to me. You could work in a lab. You could become a chemistry teacher, which, I didn't think I was interested in teaching at the time. So then I thought, oh, okay, that, it doesn't, I don't even want to work in the, lab after this. And then at the same time, in my chemistry class, half of my class are chemical engineers. And then, so I was like, oh, chemical engineers. So I looked into the careers of chemical engineers. All of a sudden, it's just like, oh my gosh, the jobs look so much better as a chemical engineer. So after my first year at Berkeley, I decided, okay, I'm going to do chemical engineering, because there's a lot of overlap between the two curriculums. But so I started studying chemical engineer, to get a real job after I graduate. But I really didn't know what chemical engineering was. As an undergrad, we didn't have a Chemie 101 to kind of tell us what chemical engineers do or what kind of problems you can solve. So I really started studying chemical engineering without really knowing what kind of jobs chemical engineers would have. And I remember during my senior year, I walked over to the chemical engineering lab, the lab that I was talking about, and I saw distillation towers and I saw this and that. And I was just like, oh my gosh, I don't want to do that. I don't want to have to go operate a distillation column. And I knew, chemistry still was my passion, and I also liked biology. And I kind of knew biotech was, back in the days, was really new and shiny. So I knew that, biotech is what I wanted to do. And for that, you have to go, graduate school. So I couldn't really make a decision about getting a job after chemical engineering, because chemical engineering, most of the students were going to working at chemical companies or petrochemical industry, refineries, and I didn't really see myself doing that. So kind of by process of elimination, I went to grad school. And then I would have to say towards the end of graduate school, it's like, I could go work for a pharmaceutical company. I did an internship as a graduate student at a pharmaceutical company. And again, I was like, oh, I don't know if I like this. Working for the company bottom line, I guess. I don't know. Those may be kind of naive notions. But at the time when I was looking at what, even chemical engineers with a PhD do, it didn't seem so attractive to me. And then, kind of thinking back to, what do I really want to do? I've always liked teaching, even though I didn't have too much teaching experience. I've always liked to share with people what I've done. And then my PhD advisor was very encouraging about pursuing, a career in academia, which I thought at the time, oh my gosh, that just seems so hard and impossible. But once you break it into smaller steps, it becomes feasible. So it's like, okay, so becoming a professor, I think I will like that, because I've always liked being in school. But let's break it down into smaller steps. Find a postdoc first, and then apply for a job, et cetera, et cetera. So I think kind of by process of elimination, I didn't really like the alternatives. Still like
so much. Thank you so much. So any other questions for Dr. Chi?
Actually, I do have a question.
Yes.
I wanted to know what was your experience like being a researcher and in academia in general, being a woman in STEM.
I would say it's been overwhelmingly positive. As a chemie in undergraduate, I think we had about 30–40% female students. And graduate school in chemical engineering, there was also, if not close to 50%, I would say the percentage of female graduate students is also high. In academia, the percentage unfortunately is a lot lower. I would say at about 10%. What do you think?
I don't know, our numbers, at least in electrical and computer engineering, at the undergraduate and graduate population, they are very much lower than what I hear. But I guess it's a different field.
It is higher in chemie, but that number hasn't changed, right?
Okay, that's
so
good.
But when you get, the percentage just drops lower and lower. I think in the School of Engineering, I think we're sitting at about 10–15%.
Yeah.
So far, I would say as a student, as a PhD student, as a postdoc, those were really good experiences. As a professor, it's been great. I love my job day in, day out. I love the students I work with in my class, undergraduate classes. All of that is great. Yeah.
That's great.
Do you have more specific questions, Ari?
Not that I can think of right now, but thank you.
Mm-hmm.
I'm impressed. Your numbers actually. So if I can tell you my story. When I was, at high school, we take a national exam in order to enter the university. And I was thinking of chemical engineering, but back then, at least in Greece, the job openings in the field of chemical engineering, they were very limited. So, but chemistry was really like, I guess it's more, maybe I could call it like more female friendly, because the numbers, the percentages that I think you said 30–40 almost percent. That's quite high. I mean, in electrical and computer engineering, the numbers are much lower. And I remember myself as an undergraduate student, I can say that we were like 5–8% female students in the year. So the numbers are quite lower.
Yeah, so chemical engineering, in our department, we have different concentrations. Bioengineering has really been, it kind of came out of chemical engineering, and it's been growing both as an industry and also, just as an area. And there are just more female students interested in bio-related disciplines. That being said, I think at UNM, our program, I want to say our numbers fluctuate between 30–50%. There were years where we had over 50% of our graduates were female. Right now, I think the number is a little bit lower. But again, it hasn't changed. Particularly when it kind of filters to PhD level.
Oh, I see.
To women in academia, that it hasn't changed. I think what also has not changed, despite all of the attention that we pay to underrepresented minority groups, that hasn't changed.
I see. Yeah, this venue, I think it's a good opportunity to inspire young students, you know, to continue also in academia, because it's not an easy field. It's quite challenging, let's say, and
Yeah, yeah.
it demands quite hard work. So I think that, you know, learning out how, you know, successful and satisfied you are with the job, because it's very important to be satisfied with what you are doing every day.
Yeah, yeah.
I think that it's very, very inspirational.
Yeah, yeah. And I think, you know, that was, you know, part of the hypothesis of our research project, is that the earlier we can show students, you know, when they're trying to get through their calculus classes and physics classes, where they're going, right? You know, the problems that you can solve as an engineer. I think it helps with persistence. And I think women are very creative, very good problem solvers. And a lot of it is also in the design, right? Designing a product, designing a process. So I think if you think of it in those terms, rather than, you know, the hardcore math, the calculations, the programming, you know, which obviously is part of it. I think, you know, we feel that the earlier that we can show what they can do as an engineer, you know, they obviously need to acquire more and more skills to work out the details. The more we can keep them motivated to continue to get through, you know, those calculus classes and physics classes at the beginning.
Yeah, it shows the light at the end of the tunnel, like, you know, what we really do with all that knowledge that we get. Yeah.
Right. And then, you know, because I think a sense of belonging, and this has been borne out over and over and over again, if a student does not see him or herself being part of this, you know, engineering professional, they're less likely to continue. But if we can show them, yes, you could be an engineer and start finding solutions to problems like right now. And you know, kind of this whole idea of formation of professional engineers. That we really want them to see themselves in creating these solutions.
Yeah. I cannot agree more with you. I mean, yeah, absolutely. Okay, so let's see. Is there any other question from our audience today? Or any comment, guys? It's not necessary to be a question. Okay, so I think, Aisha, the floor is yours. You can close the whole talk.
Yeah, I guess so. So thank you so much, Dr. Chi. It was a wonderful session today, actually. Now we have someone who works who started being in a different bachelor's degree to an engineering one. This is actually an inspiration that if someone starts with something else, but deep down they want to be engineers, they will be brave enough now that, oh, I can go to a different degree now. Thank you so much for your time, for joining us today, and everyone else. So I guess I will be ending the meeting.
Thank you.
Thank you so much, everyone. Have a very good evening.
Thank you.
Thank you.